Hello everyone, and a warm welcome to the latest instalment of the NK webinar series, which is hosted today by Lumix. My name is Shaksita, and I work with the Cell Therapy event series here at Hanson Wade, and will be chairing the session today. The plan for us today is to discuss and explore the network of cellular interactions of NK cells in the tumour microenvironment. In this webinar, we'll be joined by Dr. Lowdell, who will discuss the latest data from INmune Bio and the discovery of a new mechanism of action of its NK priming immunotherapy, INKmune. INKmune is a clinical stage proprietary tumour cell line which is administered intravenously and primes NK cells to kill otherwise resistant tumours. INKmune primed NK cells have been shown to kill a wide variety of solid and hematological cancer cells in preclinical studies. We're also joined today by Rahir Reimers, Principal Scientist and Head of Validation Research at Lumix, who will be presenting the Lumix ZMOVI Cell Avidity Analyzer, which is a one-of-a-kind instrument that allows the measurements of hundreds of affected tumour cell pairs for their interaction strength. Welcome both, how are you?
Very well, and pleased to be invited.
Thank you for the introduction. Very good.
Thank you. I'll shortly be handing over to Mark and Rahir for their presentations, and following this we will open up to the audience for a live Q&A. Please do submit all questions via the questions tab at the bottom of your webinar dashboard. Questions should be submitted throughout the webinar, and we will answer all the questions at the end. We will, as I say, endeavour to answer all of the questions in the allotted time. However, if there are any unaddressed questions, we'll pass these on to both of our speakers and we'll answer them in a post-event report. This report will be distributed after the webinar, and if you'd like to access it, please do make sure you supply your email address when registering and opt in to receive emails from us. Now, without further ado, we'll get started and kick the session off with Mark, and if you'd be so kind as to share your screen and your exciting work with us.
Thank you. You hand over to me, I'll open up the sharing. Okay, good afternoon, good evening, good morning, wherever you are in the world joining us. I hope you can see your screen. I'm sure I'll get a text message if it hasn't come up. So I'm Mark Lowdell, as you've heard. I'm a CSO of a spin-out company called INmune Bio, and also a professor at University College London. So a lot of the data I'm going to show you are academic data from my academic team, and also some company data, which I'd like to share with you. Obviously, as a company, we have forward-looking statements, so I have to show that. We're a listed company on NASDAQ. So first of all, why are NK cells important? I think this is a really, really important paper from the Lancet. It's elegantly simple, and this is a study done in Japan looking at over 7,000 healthy individuals. These are just the data from the female cohort of over 3,500. And what they looked at was one day in the life of these people to see how functional their NK cells were. And some individuals had low NK function and some had moderate or high NK function. They followed them for 11 years, and those with low NK function on a single day of their lives had a statistically significant higher incidence of all types of cancer over the next 11 years, proving that NK cells are critical for immune surveillance, but more importantly, or as importantly, showing that some of us as healthy individuals have good NK function and some of us have poor. Why is that? What's the mechanism of it? And can we move individuals from here to here? And that's what this talk's going to be about. So it's not just healthy individuals. We know in a number of different cancer settings that patients with low NK function have poor outcome. And these are data from two of my papers looking at acute myeloid leukemia and myelodysplastic syndrome. And in both cases, individuals post-chemotherapy with high NK function survive a lot better than those with low NK function. And this is the most critical in terms of what you're going to hear later. In MDS, patients with low NK function have a median survival of about 18 months and no long-term survivors. Whereas those with higher NK function, and critically this was not normal NK function, it was just higher than these patients had a median survival of over 50 months, and some of them, as you can see, long-term survivors. So again, how do we move individuals from here to here? Is it a function of their NK cells, or is it a function of their tumour? And you will see it's actually a bit of both. So how do NK cells work? Well, the NK cell recognizes the tumor cell. It makes a strong bond to the tumor cell, a tight immune synapse with the correct signals to drive it to activation and then through to lysis and the death of the tumor cell. However, some patients have NK resistant tumors, or their NK cells are actually not very good. This means that they form a weak bond to the tumor cell, and in a weak bond, they form an unstable immune synapse with no activation and consequently no lysis. This is shown here relatively elegantly, looking at an NK-resistant ovarian cancer cell line and NK cells from a healthy donor. These are live tumor cells, and you can see they have a normal spread out type morphology. And here are the NK cells binding, and almost as soon as they bind, the tumor cell starts to die. It shrinks, and you will see it break up shortly as we rattle through that process. Here you can see an NK cell binding to the condensed tumor cell and this tumor nucleus, the tumor cell breaking up into its constituent parts. In contrast, if we take the same donor NK cells, but we incubate them with an NK-resistant ovarian cancer cell line, here you can see the NK cells bind very, very weakly. They're permanently in motion, moving around those tumor cells, and there's no death whatsoever. But perfectly functional NK cells, they're very good at killing this cancer cell line, but they can't kill this cancer cell line. So this is a situation where it's the tumor cell that's deciding the outcome, not the relative ability of the donor NK cells to kill. So what's the difference there? And if we look at how NK cells work, we all know about tumor-derived inhibitory signals, both from the tumor cell and from the tumor microenvironment, and activating signals that are present on tumor. And as those activating signals increase, the NK cell moves from rest through a primed and activated state to a point where it can kill the tumor cell. And I think this image here is very important. In fact, it's the crux of what we're going to talk about today. This is an acute myeloid leukemic cell from one of our patients at the Royal Free Hospital in London, and these are her own autologous NK cells killing that tumor cell. But what's very important to notice here is it's not just the NK cell binding to the tumor cell and these very tight immune synapses, but as you can see, the NK cell binds to NK cells, not only those that are involved in the immune synapse, but also other NK cells, which are third party to the killing event. And so what we wanted to know back in 2001, 2002, when we first saw this image was, is that important? And if so, why? What we knew was that as you increase the NK cell to tumor cell ratio, this is a conventional killing assay of a K562, the number of NK cells that are actually in conjugate with other NK cells increases. So this is a physiological effect, and it's partly due to the number of NK cells, what's available to bind in your assay system. What we also showed, and this is the first introduction to the cell line that we own within INmune Bio. INB16 is a myeloid leukemic cell line that we have developed and turned into a therapeutic product. And we've membrane labeled this and then incubated resting NK cells with it for 16 hours, whereupon they become primed. We call them tumor primed NK cells. But you can see this NK cell has sampled the membrane of that tumor cell and itself has picked up some of that fluorescent label. These cells carry that membrane with them, and it's part of the activation process. And what we now know is that this is a physiological function of NK cells. They do this all the time, and you'll see that later. So if you look here at this video again of these NK cells binding to this live tumor cell, 3 NK cells, a live tumor cell. And as the tumor cell dies, you can see some of these NK cells lose interest and they move away from the tumor cell. And this NK cell here is going to get tighter and tighter and tighter until eventually, as you saw in the previous image, it binds to the condensed tumor cell, and it now starts to sample the membrane of that tumor cell. This is not INKmune. This is an ovarian cancer cell line. So this is another example of an NK cell sampling a tumor cell. We're not alone in seeing this. This is a paper from a friend of mine, Dan Davis, who was at Imperial at the time, and his group, Fiona McCann, did this work. And what you can see here is that NK cells actually pull MIC-A, out of tumor cells. This is a HeLa cell being sampled, and exactly the same image that I just showed you from MB16, the NK cell has picked up the MIC-A from the tumor cell. So what that means, what does that mean? Well, what you'll see here is that that actually leads to NK cell activation. It allows an NK cell to pass that signal, that information, that tumor exposure to other resting NK cells. We've seen this in a clinical trial that we published in 2015, but here you can see it actually under the microscope. And so here is this tumor cell, which is bound to the membrane of, this NK cell bound to the membrane of that tumor cell. Tumor cell's dying. This is the NK cell that leaves the party. This is the one that you saw go on to sample, but this one is now interacting with this cell. This NK cell is interacting with that NK cell. And if you focus on this live tumor cell here, this NK cell has seen this tumor cell, it's seen this tumor cell, it's done nothing. But now that it's been educated by that process, it just barely touches, this is a 20-minute interaction at this rate, with this tumor cell, and already it's condensing and dying. So this cell has been educated, and we have and others now tend to refer to them as memory-like NK cells. They get educated by another NK cell, and it leads to tumor cell death. So what's going on there? What drives this and what regulates it? Well, as I've said, part of this is due to the NK cells of the donor, but part of it is, a majority of it is due to the tumor cell itself. And so we all know K562 is a highly susceptible tumor cell for NK killing. And here you can see if you take resting NK cells from a healthy donor at an ET ratio of 1 NK cell to 2 targets, you get low killing. And as you increase that ratio to 2 NK, 5, 10, you get increased level of killing. If you take our tumor cell line or HeLa or something else, but we'll focus on our tumor cell line MB16 here. Resting NK cells at 1 to 2 barely kill it. It's below level of detection really. But once you get to 2 to 1, 5 to 1, and 10 to 1, you see this consistent increase, just as you do with K562, but to a far lesser extent. So what's happening here? Why are these NK cells not killing MB16 to the same degree of K562? And does that matter? Well, interestingly, if you look at the proportion of NK cells in that mix that are activating in response to the tumor, although you're getting an increase in killing to K562, you get a reduction in the proportion of NK cells that are actually activated. And it's true in the MB16 setting as well. So activation and killing are not directly proportional. And I think this is probably the most important image to show you. CD107A is regarded as a readout of degranulation. So CD107A is only expressed on NK cells, apparently, at the time of degranulation. I think you'll find increasingly that the evidence is coming out to show that's not the case. It's more of an activation antigen. But here you can see exactly what I've been trying to argue for many years now. CD107A, actually the proportion of 107A positive cells goes down as the percentage of killed cells goes up, and it mirrors the activation. And it's true with MB16. So NK cells combined MB16, they can get activated by MB16 without actually killing very many of them. Why is that important, and why is it a valuable model? Well, one of the things that we've been interested in is what makes an NK cell kill, and once it's killed, what does it do? What does the tumor do to the NK cell? And obviously you've got two scenarios. It could exhaust it. This is the B scenario. Once a bee stings you, it can't sting you again, it's dead. Or it can be a wasp, it can serially kill you and sting you. So here we've taken resting NK cells and we looked at those cells. They've got very low expression of CD107A, they haven't degranulated. And if we incubate them with Dowdy cells as an NK cell, target cell, this is 100,000 Dowdy cells at time 0, and after 4 hours in the controls, there's just a little bit more than 100,000. They've proliferated a bit over 4 hours. And if we incubate them with resting NK cells, we get about 40% lysis after 4 hours, and so the total number goes down. If we pre-incubate those NK cells with K562 for 4 hours and we get the level of killing that you saw just now, we abrogate nearly all of the ability of those NK cells to kill Dowdy cells. They've become exhausted. But if we incubate them with MB16, where we also see some killing, but not to the same degree, you would predict that you'd get less abrogation, but in fact, you get more killing. So you've primed these cells, you've made these cells better at killing. So what is it about MB16 and indeed some other cell lines that are able to do this that K562 and many others don't? And so we've looked at what happens when you incubate an NK cell with MB16 or as a control IL-2. We've looked to see which genes get upregulated and downregulated. Without going into great detail, and these are published data, over 500 genes are upregulated by MB16 that are not upregulated by IL-2 or any of the other cytokines we've looked at. And some of these are really critical: CXCL10, CD70, STAT5A. CXCL10 and CD70 are particularly interesting to this talk because they're present in the immune synapse and they're part of that signaling cascade and that definition of a good immune synapse. So if we look at this again, and these data come from Kush Kamal Das at Lumix, I'm very grateful for him doing that. But what we've looked at here is the percentage of cells, and I'm sure you'll see a lot of these in the next talk, but the percentage of cells that are bound to K562 at 5 minutes, and looking at different donors. And as you can see, donor 4 binds more strongly than donor 1 and donor 2. Perhaps that talks back to that original paper I showed you of normal healthy individuals having different NK responses. And as you would predict, when you incubate the same donors with the NK resistant SCOV3 tumor cell, you get no binding at all. You can see that here. This is the image I showed you before. None of these NK cells are binding to SCOV3 with any degree of strength or durability, and that's why you don't get lysis. What about the K562 MB16 story, though? We know that MB16 is less sensitive to killing, and you get less CD107A expression than you do with K562. And indeed, as you might predict, K562 NK cells bind much more avidly to K562 than they bind to MB16, but they still bind better than they bind to SCOV3. Now we know that when we bind an NK cell to MB16, it gets a signal that allows it to be a better killer, whereas when we bind it to K562, it becomes exhausted. What's the difference here? Well, this is where the Lumix device has been so powerful for us. If we look at what happens over time when we incubate an NK cell with K562, within 5 minutes, there's quite strong binding. But as you leave that, by 10 minutes and 15 minutes, it's maximized its binding, and it's very, very strong indeed. In contrast, when we bind the cells to MB16, they bind moderately, and they never increase that level of binding. So they're forming an immune synapse, but it isn't as strong and it isn't as stable as it is with K562. And that translates to a more functional NK cell, which is very interesting indeed. So this is looking at binding to the SCOV3 cell line, again, the NK resistant cell line. Here are the resting NK cells. You activate them with IL-2, you get very, very slight increase in avidity, and these cells don't kill the tumor cell. You activate them with IL-15, you get slightly more avidity, and these cells are able to kill SCOV3. But if you pre-incubate them with MB16, you increase their avidity enormously. And you can see that here in this image. If you look at this NK cell here bound to this tumor cell, the tumor cell is moving, as you can see, considerably. And that NK cell is almost welded to it. I mean, it's remarkable how tight that immune synapse is. And as we play this out, you'll see ultimately, there we are, the tumor cell dies. So we believe that critically, it's avidity that matters. The difference between these individuals here with myelodysplastic syndrome who have low NK function actually is because they can't make it that tight immune synapse, whereas these patients can. And can we change that? Well, we can in the lab. This is in vitro. We've looked at a tumor cell line called MDSL, and we've looked at healthy donors who kill MDSL very, very well. And this is one of our MDS patients who has a very low level killing and would fall in that population here. By incubating those cells with INKmune, which is the MB16 cell line, but made replication incompetent, we can move the patient from low-level killing to high-level killing. In fact, it overlaps donor 1's killing, which is why you only see 3 dots here. We have first evidence that potentially you could change a patient's response to their own leukemia or their own tumor in vitro. And could you do that in vivo? So these are data which are not published. They are on the website, so they are in public domain. But these are the first data from the first patient treated in our first trial with INKmune. And what we've demonstrated here is that priming effect with these INKmune cells occurs in vivo just like it does in vitro. And after 15 years of developing this in vitro, this is extremely exciting. And you can see here that within 7 days of being treated, the proportion of CD69 positive NK cells in the peripheral blood of this patient almost doubled. The proportion of cells expressing NKG2D increased, NKp46 increased. And this activation occurs across multiple different subsets of NK cells. The CD16 positive cells mediating ADCC activate, the 16 negative cells activate, the NKG2D cells activate, and the NKG2D negative cells activate, and similarly for NKp46. So what we've demonstrated here is that we can modify the immune response of a patient in vivo just in the same way as we were able to modify it in vitro. So in conclusion, NK cells interact not just with tumor cells, but they activate with NK cells. They interact with NK cells. And the signals that they get, both from the tumor cell and from NK cells, are critical for their function, and they affect hundreds of genes in that process. And we're only dissecting those slowly. I think most importantly, degranulation, as evidenced by CD107A, is not a direct correlate of lysis. And so you can see, as I showed in those earlier slides, relatively low CD107A expression, but high killing, and in contrast, high CD107A percentages and relatively low level lysis. It also therefore means it's not a measurement of exhaustion. So CD107A positive cells, and I didn't share those data with you, but if you sort the CD107A positive cells out of the population, those cells can quite often go on to kill again. And once again, we believe it comes down to which of those CD107A positive cells retain the ability to form a tight and stable immune synapse with the tumor cell. We also know that tumor cell conjugation may activate an NK cell without triggering it right the way through to lysis. And that's what we think is probably happening when the NK cell binds to MB16. Some of the NK cells kill the MB16. We see that 20%, 30% lysis. But many of the NK cells plainly don't kill MB16, but they've had enough signal to take membrane from MB16 and to use that to drive them from a resting state to an activated or primed state. In fact, we know that we can use membranes from MB16 to cause activation of NK cells without them ever seeing the intact cell. We also know that this increases the TNF alpha and interferon gamma secretion by these cells. So although I didn't show you the data today, they not only increase lysis, but they increase their ability to produce effector cytokines. And this is all associated with an increased avidity to tumor targets. So potentially you could use avidity to determine whether your patient is responding to the drug. One of the problems we have with all of these adoptive cell therapies and immunotherapies is they're expensive, and targeting the right patient is critical. And so if you can take NK cells from a patient into the lab, incubate them with MB16 or INKmune, and look to see what that does to the avidity of their binding to a target cell, it gives you a biomarker before you even select the patient. So when we get into our phase 2 trials, almost certainly we will be doing that. We will only select patients who we know are very likely to respond to the drug. That reduces the cost of your trials. It increases the likelihood of a signal, whether it be positive or negative, but it does at least increase the likelihood of a signal. And so we believe that the ability to measure avidity is going to be very important in NK cell biology, just as it's already being shown to be important in selecting the right CAR, for example, for a CAR T. And increased avidity increases the lysis of the cell, but interestingly, it doesn't change the duration of the conjugate. So the NK cell binds to the target cell for the same length of time, but just better. And so we believe that we can use this as a tool to develop better NK cell activating agents. So as we've seen with every drug that's ever been developed over the last 20 years, there's always son of drug, daughter of drug, grandson of drug, great niece of drug. So what we believe we can do is use this to develop the next iteration of INKmune and its other cell types to better activate NK cells, and indeed mixtures of the cell with cytokines, for example. So you'll all be aware of the drug trials with IL-15 receptor agonist and its like. So this gives us the first opportunity, I think, to look at the true biology of the interaction of the tumor cell. And ultimately, I suppose our next step is to look at the avidity of the binding of the NK cell to other NK cells and how important that is in the true biology of NK cell function. And what we've just started a program at the moment is to look not only at the molecular basis of this increased avidity, and you've seen some of the data on CD70, the CD70-28 interaction, the synapse is critical, CXCL10, it's important in getting the NK cells into the tumor. But also looking at the proteomics, the complex proteomics. I think single cell proteomics is going to radically change this field over the next few years. So I hope that's inspired a lot of questions, and I'm happy to take any of them at the end of the session. But I have to, of course, acknowledge all of the people that have done this work. And I said, I'm not just CSO of INmune Bio, I'm a full professor at UCL, and so many of these are my UCL staff. But I must thank Aga, Xenia, and Elena predominantly for the data that you've seen, Kush, without whom we couldn't have set up the MOVI system, and my fellow colleagues at INmune Bio, they sit back in the office and make all this happen. Thank you very much.
Thank you so much, Mark, for that wonderful presentation. We'll move over to Rahir for his presentation now, but just a reminder, do keep submitting your questions. We've got a few already and we will take them at the end for our live Q&A. But as I say, for now, I'll hand over to Rahir for his presentation.
Yes, thank you. Let's see. Is it up right now? Yes, okay, excellent. Yeah, welcome everyone. So thank you for joining this joint webinar. I think Mark has a very inspiring talk with a prominent role of cellular avidity for NK cell selection. And today I would like to go in a little bit more detail on how you can actually measure cellular avidity for your immunotherapeutic products with our latest technology, the ZMOVI cell avidity analyzer. So I think NK cells as a living drug still is something that needs optimal preclinical design and selection criteria for NK cell therapy. So it's still a work in progress, also clearly stated by Mark. I mean, there are functional differences of NK cell sources, and this is still unclear why that is. Autologous NK cells have several difficulties, including manufacturing time and quality of the product. And use of allogeneic NK cells often results in rejection 2 to 3 weeks after infusion, which is also a huge problem. And NK cells only last shortly, and they proliferate poorly. And there's a plethora of activating and inhibitory receptors that determine activity and efficacy. And all these things combined can result in non-curative NK cell products that could fail clinical trials. So I think in general, with cell therapies, we are looking for the immunotherapy sweet spot. So we would like to have low exhaustion, no possibilities for antigen escape, but also low on target of tumor toxicity. You would like to prevent neurotoxicity, cytokine storms, while you would like to increase the persistence and the function and the penetrance of your immune cell products. So you would like to prevent toxicity and relapse and really have a cure only for patients, where you actually also need to tailor the therapies to the patients specifically. So best information should really result in best therapy. So we believe that there is a need for a better design of methodologies to achieve optimal anti-tumor responses. So an illustrative example here of how cellular therapies could improve is shown here. This is data obtained by the lab of Martin Puhl at UCL, and here they use two different CARs, the FMC63 clinical CAR and a CAR that is an analog of that but had a 40-fold lower affinity. It's called the CAT CAR. And interestingly, what you see that at every E to T ratio, this CAT CAR is actually doing outperforming specific lysis of the target tumor compared to the high affinity CAR. And also, and maybe even more striking, if you put these two CARs into a mouse model, you can see that the high affinity CAR is actually able to slow down the tumor outgrowth, but the low affinity CAR is actually a CAR that is also that is very significantly reducing the tumor burden. So here is a clear illustration that you don't need the highest affinity, but you need an optimal affinity for your cell product to perform best in vivo. So in essence, the traditional biophysical methods like SPR primarily focus on affinity. So one ligand and one receptor cell pair. This type of interaction completely overlooks the biological complexity that is linked to cell function. So what is missing is cellular avidity that can be described as the overall strength of interactions between a diversity set of receptor ligand pairs at the cell-cell interface, as shown in this figure here, which was also described by Mark, where these immune cells form a immunological synapse with the cancer cell, and this is very important to execute their function. So not only are the CAR and the TCR binding to the tumor target, also many other interactions must be considered. So because we know that anti-tumor response is really driven by these overall interaction that takes place at the cell, the immune cell tumor cell interface, it is the T cell binding avidity that drives the tumor killing through the formation of a strong immunological synapse, which can be formed upon the initial recognition of the target. And in this specific synaptic state, the interaction forces substantially increase because of the active recruitment of adhesion molecules. And only in this state, the T cells can execute the killing very efficiently by secreting these cytotoxic granules. Mark touched upon this, but I think the audience is well aware that NK cells express a high amount of both activation, stimulatory molecules, as well as inhibitory receptors. And there is a balance between forming a synapse or a so-called kinapse. So depending on what type of interactions these NK cells form, they either choose to form a synapse and kill the target, or they do just the immune surveillance and they are inhibited to kill the tumor. So to measure these kind of interactions, so the collection of interactions, there's a need for technology that allows direct fast and high throughput measurements of cellular avidity of individual cytotoxic effector cells. A short history of Lumix. So we started our company in 2014 and developed different instruments, but the one we are discussing today is the ZMOVI cell avidity analyzer, which we launched last year, April. And we have now already installed several of these instruments, both in the US and in Europe, already resulting in peer-reviewed publications as shown here, where for a CAR T-cell product, highest avidity correlated with highest tumor cell clearance in a mouse model, where they show that the in vivo differences, and this was in the lab of Eric Smith at MSKCC, that the in vivo differences between the vectors were not predicted by in vitro assays, but they did correlate by the cellular avidity measured by ZMOVI. So ZMOVI quantifies the binding strength directly and in a high throughput manner at a single cell level. So it's predictive for the effector cell function. It's a reproducible method because we use a biophysical way of separating the cells, which I come into within a minute. And your results can be acquired within minutes. So here you see our technology on the top, where you see the ZMOVI chip. Within this chip, you have a small microfluidic system where you have a lab on a chip, and on top of the chip, you have a piezo element. And this piezo element will induce acoustic waves. And if the magnitude of the force exceeds the binding strength of the cell pairs, the effector cells will be lifted into an acoustic node, which can be considered a zero gravity area where the cells float until the piezo is switched off. And here at the right, you can see a movie of how this looks like when you run a real experiment. So here you see fluorescently dyed effector cells in a chip randomly scattered on top of a non-fluorescent monolayer in black. And when acoustic force ramp is started, the effector cells are quickly separated from the monolayer and accumulate in these acoustic nodes. In this field of view, you can measure the avidity of up to 300 to 400 cells. Here you see a simplified overview of the workflow, which I will be happy to go into detail if necessary. But basically, you select your target cells, which could be a panel of cells expressing different levels of antigen, which you flush into the chip and let them get stuck to the surface. Then you prepare your effector cells and dye them with a red fluorescent dye for tracking, as I showed you in the movie before. And finally, you incubate the effector cells with the target cells, which takes about 5 to 10 minutes, followed by a 2.5 minute force ramp going from 0 to 1,000 piconewtons. And after the force ramp, you can wash away the effector cells and incubate your next effector cell population. And in this way, you can do 5 runs after one another on one chip using a single monolayer. This is all it takes to perform several runs per chip, and subsequently the data that has been recorded can be displayed as avidity curves shown here on the left, or as bar graphs shown on the right that show specific binding compared to a control, which can be gated, shown here in this gray area, separating specific binding and non-specific binding. So how sensitive is this system? So what you see here, you can see 3 avidity curves of 10 repeats each, divided over 3 chips of TCR engineered T cells. The gray curve is that of the control, and blue and pink are curves of TCR engineered T cells that are specific for the target. And when gating for non-bound cells, we found that 2 different TCRs for the same target and a similar HLA restrictions show distinct binding profiles that could not be determined by gold standard assays like cytokine secretion or killing, in vitro killing. When shown in bar graph, you can appreciate that using the ZMOVI technology, you can significantly detect small avidity differences between different T cell populations. And here you can see an example for CAR T cells. Wild type EuCAR T cells were mixed with EuCAR T cells expressing a CAR, and cellular avidity was measured for the same target monolayer expressing the target antigen. So what you can see, if you have a 100% pure population of transduced EuCAR cells, you see a very high binding. And if you titrate in these negative CARs, negative EuCATs, you can see that the avidity to the target is diminishing to almost background binding, shown in the lightest color. What you can see here is the bar graph with very significant differences that can be observed using the system. So we are currently use this EuCAT CAR system as a so-called model system to address avidity-related questions. So wrapping up here, I would like to finish with summing up results that have been obtained using our instrument and something to let sink in. So CARs and TCRs do not need the highest affinity for best performance. So what I'm summing up here is proof data that we have, and we are happy to share if you're interested. CAR T cells require a suboptimal cellular avidity, while TCR T cells do best having the highest cellular avidity. Measuring avidity kinetics suggests a predictive value for persistence in line with clinical findings. Avidity measurements can be readily applied to different effector cell types and T cell engagers as well. So we use T cells, NK cells, bispecific antibodies, and we are also moving towards B cells. Avidity measurements can be extended to cell ligand interactions. So not only can you do avidity measurements by coating the surface with target monolayer cells, but you can also use ligands only. I think most importantly, the first studies reveal correlation between avidity data and in vivo tumor reduction and cell function. And as Mark nicely illustrated, most potent NK cell donors can be identified with the technology. So we believe, as Mark actually also stated, that cellular avidity has the potential to become a key quality attribute used for cell product release to patients. So with that, I would like to thank you for listening, and I would kindly invite you to visit our website as well for further questions, or if you would like to set up a demo in your lab. And I would like to give the word back.
Great, thank you so much, Rahir. Just before we move into the Q&A, we have a quick poll that we'd like to share. So this will pop up on your screens shortly, and we'll take a few seconds to complete that now. Great, thank you so much for doing that. If I could now start the Q&A. We have a whole host of questions for both of you. So I will just go through them one at a time, and I'll give it, I'll make sure I say who it's directed to, but also if one of you has a comment you'd like to add as well, please feel free to interject before I move on to the next question. So the first one we have here is for Mark, and the question is TNF alpha and interferon gamma a better marker of NK cell activity than CD107A?
That's a very good question. It's certainly a very critical markers of NK function in terms of cytokine release, but NK cells can release cytokines. Different subsets of NK cells do have different functional roles. So yes, you will identify a population of NK cells that are functional with cytokine release assays, but you'll miss those which are mediating cytotoxicity by another route. So we always combine them. We do a cytokine release assay, and we have either a flow-based assay for cytotoxicity, where we look at absolute numbers of live target cells, as you saw, or we have an Excelligent system as well, which we're very pleased to use on adherent cells, which gives you a sort of what the MHRA described as a mouse in a plate, because you've got dynamic NK function as the tumor grows over 5-7 days. So we combine both, but yes, I think CD107A probably isn't a very good marker of cytotoxicity per se.
Okay, and a question here for Rahir. How does binding avidity affect the safety profile of the cell therapy, whether NK or T cell, and i.e. risk of CRS?
Yeah, so it's a very important question, I think. What we can say right now is that so, for example, for CAR T cells, where we find with highest affinity, we also identify the highest avidity, so the binding strength is there also highest. What we know for FDA-approved FMC63 is that it can induce a very significant amount of CRS. But if you down-tune this affinity, where you can also see when you use Zimovi avidity analysis that you also lower the avidity, this could be an indication that also decreasing the avidity for the target could have an effect on the safety profile for these CAR T cell products, for example. We believe that this can be very important, and it should be explored in more detail, and that is actually what we are currently doing with a collaboration in the United States.
Okay, and a quick follow-up question here, I think that relates to that as well. Is there a threshold of avidity that defines better NK function?
That's an interesting one. Maybe I could also ask that to Mark, but I think for NK cells, it might be the same as what you can see for TCR T cells. So if you engineer T cells to express a TCR, there you need actually the highest avidity for the best function. I feel that for NK cells that also actually don't show any cytokine storms, for example, in the clinic, likely you will need highest avidity there as well, but maybe Mark would like to say something about that as well.
Yeah, so I think we have to dissect the difference between cytokine storm risk and cell type and cell function. So avidity is critical, a critical part of the cell function. But what that function actually ends up being is determined by the type of cell. So NK cells don't produce pro-inflammatory cytokines, and even giving huge numbers of NK cells in the presence of low-dose IL-2, we've never seen CRS in any of the studies. So the idea that NK cells can initiate CRS, I think, is pretty fanciful. In terms of CRS from CAR T cells, yes, the degree of activation is going to be critical, but also it depends upon the makeup of the CAR T cell. Nearly every CAR T trial that's been done has been done with autologous CAR T, and I think we've got 11 CAR T trials running at the moment. I don't know how many CAR T products we've made over the years for clinical trials in my academic setting with Martin Poulet's group at UCL, as you saw those data from. But it's hugely dependent upon the type of product that you give. And so if your CAR T actually has a large number of pro-inflammatory CD4 cells, then you're going to get a higher risk of cytokine release, irrespective of what the degree of avidity is for the target. So I think it's a much more subtle scenario. Actually, there are too many complex interactions going on. It's mediated by too many different variables. In terms of threshold for NK function, I think that's really interesting, and we would love to get enough data to show that. The data I showed you are some very preliminary data, and the issue there is that the degree of avidity for killing some tumors is necessarily lower threshold than others. So there's and we've not standardized that. So we've not been able to say, well, okay, we've got 5 different tumor cell lines, and we're going to standardize the avidity signal from those. And then once you get above X%, that leads to lysis. We can say for something like SCOV3, if the avidity goes above 25 or 27%, I think it is, you get good killing, and if it's less than that, you don't. So we are getting those sort of data. And when it comes to a therapeutic product, obviously INKmune itself is not an NK cell. It's what we call a pseudokine. It works like multiple cytokines. It gives the activation signals in vivo. But in a past life, I was doing adoptive NK therapies, and there you would want to know how good your NK product was, just like we want to know how good our CAR T products were. And that's where I think the threshold avidity will be very important, because as we all know in this field of manufacturing cell therapies, potency is one of the big challenges. How do you show that your product is going to be potent? And I think avidity is going to be a really, really key tool in determining the potency of both CAR T cells and indeed adoptive NK therapy products. I'm sure that's what Glycostem are doing, at the moment, given that they've also got the ZMOV in their facility.
Great. Another question to you both, actually, that I think sort of links with what you're saying there, Mark. Do you consider NK monotherapy to be a promising approach for solid tumours, or is the big effort to get combinations with therapeutic antibodies?
Now that's extremely interesting. So therapeutic antibodies are something that obviously, rituximab, Herceptin work through NK cell binding, and they're very successful. There are failures, both. And that could be because the NK cells in those patients aren't great, in which case you've either got to make the NK cells better, or you've got to give an adoptive therapy. So I think that's a scenario where an adoptive therapy works very well. The problem is that when you grow NK cells in the lab, or activate NK cells to grow them, because as you heard, they don't proliferate well, what an NK cell does when it activates, it synthesizes and ADAM17, which is a matrix protease. And ADAM17 cleaves CD16. So you immediately take an NK cell that can mediate ADCC, and you render it into an NK cell that's very active, but it can't mediate ADCC. And you've seen out there are drug trials of cells that are genetically modified to enhance their CD16 expression or express high affinity CD16 or ADAM17 resistant CD16. So I think that's where the challenges are. It's too simplistic to think that you can just make lots of NK cells, give them to a patient, throw in a bit of rituximab or targeting monoclonal antibody. I think what's probably more valuable, if you use an antibody, you're targeting one antigen. NK cells are fantastically designed to target multiple antigens, MIC-A, MIC-B, all of the ULBPs, and whatever else we find out there that are ligands for NKp30, P46, etc. So using natural cytotoxicity receptors on the NK cell is actually extremely valuable. And therefore, being able to give NK activating agents like IL-15 receptor agonist, like INKmune, or combinations thereof, actually are probably more likely to give an outcome than giving combinatorial therapies with antibodies at the moment, until we've solved the CD16.
Okay, great.
Yeah, so I think the only thing I would like to add here is that probably one therapy to rule them all is, I think, not the likely way to go, or how it will happen. You see more and more therapies also focusing on combining therapies, and I think that's probably the way to go. But also, of course, patient-specific treatments, which is very important. And that's something we are also trying to consider how we could analyze that using our technology. And I think that's a big challenge, but it would be nice, for example, to see how the T cells actually interact with tumors within patients. So not using cell lines, but for example, biopsies, and look at the more heterogeneous character of the tumors and how these T cells respond. Because it could very well be that you have a hostile environment of the tumor where the cells are inhibited, and it would be very interesting also to be able to measure that using avidity here as well.
Great. We have quite a few questions on avidity here, but There's another one here around, can NK tumor cell interaction prior to infusion in patients enhance cell migration or homing to solid tumors?
I would love to think so. I have no data to support that. Well, actually, we've published, this is all I can say to that, and it's not a solid tumor, but it is homing to a tumor microenvironment. So in our paper in 2017, where we used the precursor of INKmune, so grandfather of INKmune, we activated donor NK cells ex vivo. So we did the sort of Jeff Miller haploidentical NK infusion after a bit of Seattle conditioning. And this is in patients with AML. And one of those patients, we were very lucky in that he responded well to his first infusion, and then his disease relapsed. And contrary to the trial, we were allowed by the MHRA to give him a second dose. They approved protocol amendment over a weekend, actually. We gave him a second dose. So these were haplo-mismatched NK cells with an HLA disparity that we could follow by flow. And we looked in his bone marrow at 48 days after he'd been given the cells. And the 33% of the NK cells in his bone marrow were donor NK cells that had been activated ex vivo by the MB16 parent line. None of the NK cells in his peripheral blood were donor. So they had tracked entirely to his bone marrow, which was the site of his disease, and engrafted there, it seemed, actually for quite a long time. He remained in remission for another 11 months after the infusion and second dose of cells. So yes, I think if you give the cells the right signal, they will go there. I also think in terms of tumor infiltrating lymphocyte data, I think a cell that's taken out of a tumor and activated, expanded, whatever, has a higher chance of going back to the tumor because it's been pre-educated and it's got adhesion molecules, etc., than taking the same cell out of the peripheral blood and trying to activate it. But those data will come out shortly because I know Tom Schumacher is doing those experiments generating antigen-reactive cells from the peripheral blood that are reactive to the same tumor antigens that the TIL cells are. And so we'll see what comes out of that. But yes, I think there's a very good chance that, for example, the upregulation of CXCL10 we've shown, but we've also shown CXCL4 and CCL6. So yes, you could imagine that if you activate a cell with the tumor, it's going to activate adhesion molecules, and hopefully that means that they will track in vivo.
Great, okay. And we'll take this final question, again, open to you both. What is the best cell source to develop allogeneic NK cell therapies for solid tumors? iPSC, placenta, cord blood, HSCs, PBMCs, etc.?
Yeah, maybe I could start as being not the expert, but I think there's the clear, it's probably also the same holds for perhaps T cells as well. So where do you get them from? Do you induce them from iPSC? For NK cells, do you get them from cord blood or peripheral blood? I think the source is very important. Also to obtain sufficient amount of cells, the cells that are fittest. But I think for now, each one of them has their benefits and their disadvantages. So I think we're still not entirely sure what the best source is. And then I will give the word to Mark, who will probably have more smarter things to say.
Not smart, but just a bit more opinionated, I suppose, which is what I'm famous for. I think the data coming out of the iPSC trials at the moment are suggesting that an iPS-derived NK cell may be different, whether that's good or bad, but may be different to an NK cell that's derived from peripheral blood or cord blood. I do particularly like cord blood as a solution. I'm not a great lover of cord blood NK cells because of the limited number, but to derive NK cells from a cord blood hematopoietic progenitor, we've published that, and Glycostem have it in clinical trials at the moment. I think that's a good way to go because you've got a defined population that you can work with, and you're not pushing the physiological boundaries of the cell in the same way that you are, at our original trials using haploidentical adults. Great on a patient-specific basis, but very, very challenging to deliver, and I wouldn't go down that route myself. So yeah, cord stem cell derived NK cells are my bet at the moment, but I've been proven wrong many times.
Great. Well, thank you so much for both joining us today, for giving your presentations and also for this live Q&A. Thank you to everyone for submitting those questions. What we will do, we've had a load of questions, so there's still lots that we didn't get through. We will, like I say, submit these to the speakers, get the answers and put those together in a post-event report for you. If you do want to receive this report, please do remember to ensure that you have opted into our emails and have provided us with your email address. If anybody does have any follow-up questions or comments, feel free to please email these to myself and I'll make sure the speakers get them. It's shaksita.desai@hansonwade.com. That's shaksita, SHAKSITA .desai, DESAI@hansonwade.com. The recorded webinar will be shared with you all shortly, and along with our post-event report in an email. Thank you all for your time today, and I hope you enjoy the rest of your day.
Thank you.
Thank you, everybody. Thanks for the questions.